I'm thank Dr. Abhishek for giving me this opportunity. And as you can tell from the gray hair, I've been around for a very long time. But I did not break any norms. I actually obeyed all my parents and everything and did what was right. We looked after in-laws. I have a daughter who was born before my PhD. I got married during my master's because my mother said, if I got married, did a PhD, nobody will marry me. So she will be stuck with me. So I had to get married. So everything is fine. We have to do what is required of us. That is our dharma. So we have to do that and then we can proceed in life. So today I would like to show you two examples of something that we have already published on uh, machine learning applications. We also do other kinds of bioinformatics studies, but since this is a data science conference, this is important to share with you how we can actually choose data and how we can choose features that will help us to interpret the results. Because if we just did unsupervised learning, we do get excellent results, but how on earth do you figure out what has contributed to this good result? And when you make a prediction, you need to be able to tease out the contributing factors. And it's only for that that we actually use features. And once we have the features, we have explainable machine learning, explainable AI, and therefore that is easier to interpret, especially when you work with biologists or clinicians or other biological scientists. And we would like to understand the contributing factors. So that being the case, let's share the screen and get on with our presentation for today. Okay, so machine learning applications in bioinformatics, and I put my story here. So I live in Sydney, Australia, and I've lived here for some time now. We left Delhi because I just couldn't live in the pollution at that time. So having a lot of issues with my lungs, we had to move. But Sydney is a beautiful place and hopefully you will be able to travel once all the travel bans are lifted. I believe India is now open for travel. I think Chennai has opened up, my family tells me. We are here under lockdown, so we have to do everything on Zoom. So I'm stuck to my chair, and after giving one lecture, I'm here with you to give the talk today. So here are two applications that we are going to talk about. One is finding native protein-like models so that we can understand how we can use machine learning to interpret protein 3D structures. And at the same time, we have done some chemistry-oriented work, which is very significant for drug design. We are looking for bioactive ligands for human olfactory receptors. So these are two different topics, and I will take you through them to show you what can be done, what is possible, what are the limitations, how do you choose a model, how do you choose a methodology, and what data do you require to do your work efficiently. It's all a matter of trial and error, and you need to also read enormously to figure things out. Science is actually one entity, and we have ourselves put and divided it into pieces, almost like a pizza slice, and we say this is physics and that is maths and this is statistics. It's actually one. So that is why we are able to move from one area of science to the other, and we should be aware of what our colleagues are doing and developing in the other areas so we can enrich ourselves and use their techniques effectively. Mathematicians decided on graph theory. A long time ago in the 1700s, Euler came up with graph theory. We used it to understand alternative splicing, and we made a number of graph databases way back, I think in the early 1990s. So there are ways to apply these things, and that's why we need to keep an open mind how we can actually harness developments in other sciences or engineering and use them for our data, which could be from medicine, from clinical samples, from images, which is image analysis, which is also used to actually sequence DNA, and in bioinformatics to understand molecular issues. So here we go. A little bit about me. I actually studied in India and I won a National Science Talent Scholarship and studied my master's and my bachelor's in Chennai. My PhD, I moved to IIT Delhi because, as I told you, I got married and had to move to Delhi, but I did my PhD there. And then I moved to France for a postdoc. At that time, I had a 2-year-old daughter who went to stay with in-laws so I could go to France. So we did the sacrifice bit, but everything is fine. We are back together now. So then I did a small postdoctoral fellowship in the US. We were wondering at that time whether we should move to the US, but then we loved India. We wanted to be in India. So we came back, worked hard. I was a lecturer, leader, and then head of department of chemistry at Maitreyi College at University of Delhi. But it was teaching, teaching, teaching all the time. So I got a little fed up. I was information scientist at the Bioinformatics Center at JNU in New Delhi for 2 years. But by then my health was deteriorating. So we moved to Australia to get away from the pollution. So now I've been here since 1993. It's a long innings. I came as a research fellow and then continued on. And then I worked for a national service organization like NCBI, which no longer exists because Australia decided to close it down. Then after that, I went to Singapore. And that was the breakthrough. Going through the glass ceiling, I actually got an associate professorship. I'm still very fond of Singapore. We spent 3 years there. And then I decided to return to Australia because I got the first chair of bioinformatics and I've been with this university since. I do whatever I can where is required. It's not that everybody can't do it. Some people put in more effort than others. And we also still make dinner, iron our clothes, do all the housework, do all the gardening, we do everything. So it's just a matter of organizing our time and being a productive person in science as well as at home. So what is machine learning? Algorithms can actually predict new instances based on the data they are given for learning. Learning is what we normally do when we go around. When you go or walk to your school when you're a child, you figure out that this is shorter path than another path. So it's just a way of learning. And when you have different types of data, you can carry out different types of learning. Now these learning methods have actually got names like decision trees, support vector machines, regression, naive Bayes classification, hidden Markov model, random forest, recurrent neural network, short-term memory, long short-term memory, and convolutional neural networks. Some of these are very popular in bioinformatics today. In our current conference in COP 2020, we have received 4 papers on convolutional neural networks, which I've just assigned. And of course, we also see other methods. We have used ourselves random forests and some of the others, and we will show you how they match up and which ones work on which data. There is no rule in this. You have to try different things, and usually you take your known data and split it into 2. So while there are many, many types of applications possible, and this I borrowed from briefings in bioinformatics, which had a very nice overview of machine learning in bioinformatics, we are presenting to you structure assessment and drug design as 2 of the potential applications. So in our structure assessment, what was the problem? What were the methods and what did we do and how did we do? That's the question. Protein structure has always fascinated scientists because proteins fold in weird and wonderful 3D structures. For this, you need to be good at 3D algebra, 3D geometry, and 3D vectors. So that's how we decide which atom is where. So you need to understand 3D space. So it's really exciting. Only now we have 3D printers. We had to imagine all this in our head. So for us, it was a big learning experience. The reason we are interested in the structures is that although we have millions and millions of proteins, they seem to form relatively limited number of three-dimensional shapes. And this is encapsulated by almost 1,000-fold decrease in the number of protein forms that we normally see. And the structures are also about 1,000-fold less. So these are all in millions. And as you can see, there are already 3 numbers here, whereas here we have zeros to give you the difference and what's happening. So although we know that proteins fold automatically when they are being expressed by the ribosome, we really don't know what is the little hidden secret which tells them, hey, can you please form a helix, or can you form a sheet, or can you form a combination of both? And this is still not known. So it has remained the holy grail of protein structure prediction. So this is one of the big challenges in computational biology and has been for many, many years. So predicting the 3D structure of a protein given only its sequence is still challenging, and still people are arguing about it because there seems to be no way we can crack what nature is doing, and that is the challenge. The best available method is when you have a relative which has a structure which is called homology modeling. We use that template or the 3D architecture to build a new related structure. And that gives excellent results if the structural homologue is available. But when there is no structural homologue available, we do know that structure is conserved far more than sequence. For example, we have the TIM barrel, which is the triose isomerase protein, and that fold seems to recur in totally different circumstances. Something like the wheel. So the wheel is available for the cycle or the tricycle or the aeroplane. So there are different variants on the theme. So this kind of structural evolution has occurred for many millions of years. So we can use that as one of the ways to build a model and then see whether that model fits that particular sequence and evaluate it. Maybe that will help us to understand better how to predict a 3D structure a priori. The next best method is therefore to take this fact that structures are conserved, take all the 3D scaffolds, take away their side chains, and simply build thousands of models and then evaluate them using energy functions, usually repulsion, attraction, van der Waals, etc., dipole moment, etc. And then you evaluate and then you see, okay, this one looks a bit better than that one. So I'll throw away the ones that are high energy, keep the low energy ones and come up with a model. Here we do well when we are looking at individual domains, which are sometimes subparts of the big protein and which actually fold uniquely on their own. And they sometimes are associated with a unique function. So I think Saudamini, who's coming after me, is a great domain expert, and she will be able to explain to you much better than I am. Okay. The next best method would be, if that one fails, fragment-based 3D structure building. This actually Professor Jairam from IIT Delhi has mastered, and he uses this in his CASP approach, in critical assessment of protein structure predictions, where they use pieces of proteins overlapping with each other, look for their individual structures, and then try to pull up little bits of Lego together and build your 3D structure. Again, we need to evaluate and then validate, and then we get the final structure. Combine the fragment templates, minimize them, and then hope that things work out. The least accurate of them all, when all these ideas fail, is to simply take your unfolded protein sequence and start shaking it up, doing dynamics and hoping that it folds. This was the life work of many scientists, especially from the chemistry side, where they were using force fields to evaluate protein folding. And this has been going on for centuries. Now, when structure prediction methods were very popular, that's about 10 years ago, almost 20 years ago now, 1994, scientists got together and said, we need to actually see how these methods perform. So let's have a competition. So that is called CASP, Critical Assessment of Structure Prediction. And about 20 years later, we have something called continuous evaluation of structures. CASP occurs every 2 years, and that was the first competition to be started. People who are solving structures experimentally, usually using X-ray crystallography, provide only the sequence to CASP, which is then provided to predictors, people who have methods or who have some programs that use known methods in an optimized fashion to make a structure. They submit their predictions, and then all is revealed. This is like a double-blinded trial. So when everything is revealed, the structure comes out, the predictions come out, and a team of experts evaluate the predictions, and they have a meeting. This year, I think the meeting will be virtual. The CASP just got over, and there will be a meeting. I'm sure assessments are going on right now. About quite a few years later, it was decided rather than wait every 2 years, why can't we just do this in a continual fashion? So that is Cameo, where you have continuous evaluation of predicted structures. Quality assessment is done by the protein structure prediction PSP community, and a lot of scores similar to the other one are developed, and this is ongoing while this is sporadic. Okay. What is it about protein folding that makes it so difficult and interesting and challenging at the same time? A native fold is supposed to be completely extended when the protein is made at the ribosome. However, it very quickly decides that this is not an energetically favorable situation and starts folding up into various intermediate states, maybe saddle points, but finally arrives at a reasonably low energetic minimum, which is called the native structure. This is not a zero energy state, because then the protein won't be functional and we will all be dead and gone. So we need something that is reasonably stable, but is able to have some flexibility to perform its function. And that is called the native state or wild type structure, WT here for the structure people. So misfolding, on the other hand, represents a state of high energy, which can therefore expose parts of the protein which are normally hidden, can cause immune response or deposition of proteins, and can relate to various disorders. The deposition actually leads to Alzheimer's and other diseases, whereas when you have exposure, you cause or trigger an immune response. So that leads to overactive immune responses, challenges, and that sometimes happens even in COVID, as you know. When people are very sick, there is a huge immune cascade that is mounted, and the body thinks that it's under enormous threat and goes into a terrible war zone. So this is similar to that, and diseases are the problem. Transition states are intermediate states. And also when new areas from the interior of the protein get exposed, you can actually have protease activity. So it's all very simple. We believe that this entire thing is driven by only one thing. Hydrophobic patches on the surface are hidden when a protein folds. So how difficult can this be? So we got a protein that was submitted to Cas12. This is of course a few years ago. And we took the target, which is called T0859. And these are the details. It's from a bacteriophage. And we calculated the hydrophobic residues which are prone to aggregate. And these are shown in red. And we used a program called AgriScan. There's no need to redo what somebody else has done, just give them credit. And then we looked at the surface accessibility by using the blue background and saw which of these are on the surface. So obviously, if they're all on the surface, then we have an issue because we are not folding the protein properly. So we wanted to see how many of these hydrophobic residues are on the surface and do analysis. So we thought if we could do that, we can actually come up with some measure. And this part is, of course, from our side. While we used other people's programs, we came up with these measures ourselves. So we realized that if 50% of the accessible area is actually solvent exposed, then it should be considered surface exposed. So its SAP score in this case would be decided by the residues summed up and normalized over total number of aggregation prone residues. And then we decided rather than have a continuum of states, we will just have simply 1 and 0. So solvent accessible surface area is considered, and we take a SAP score of 1 if it is accessible, and the rest is considered 0 and carried out statistical analysis. So mapping this aggregation propensity can actually help us to estimate whether the protein is in its native state, while much of the hydrophilic surfaces, that is those that like water, are outside, and the ones that are hydrophobic, most of them, the red parts, are on the inside. So that's what we thought we should be looking for. Whereas in a non-native or intermediate stage, which is higher in energy, we would have more of the red blobs on the outside and fewer of the blue blobs on the surface. So more blue indicates native-like, and more red indicates non-native-like structures. Okay, so we took the whole of the PDB, all the non-redundant data in the protein data bank, the structural database for protein structures, and then removed the short ones. The poor quality ones chose only structures less than 2 angstrom resolution, chose only those that had proteins, no DNA, no carbohydrates, etc. No ligands were present, removed all the ligands. Less than 30% sequence identity, that means not from the same family, so we are not repeating ourselves. So there's no duplication of structure. So we ended up with only 1,500 odd structures. Out of these, almost 1,350 had a SAP score of less than 30. So 90% of the native structures have only 30% predicted aggregation prone residues as solvent exposed. This was good news because most of them are well folded and all the hydrophobes, most of them, 70%, are hidden. So 90% of the proteins are in this region. Only 10 proteins had a very high score, more than 50. That is, their hydrophobic residues were on the outside. And then we went in and analyzed them. They were aggregation prone proteins. They were already known to aggregate. They were multimers. So if you took one monomer, then it is not so happy and it has hydrophobes, or it binds lipid phosphoinositide binding. So either way, we could show that this was good. So we thought, okay, we have a wonderful idea. Let's send it for publication. That didn't work because they wanted many, many, many structures to be analyzed, not just the PDB. Applying this to all the native structures that were predicted for CASP, we found that we could discriminate the best models in red from all the rest by only using this one simple score, which we digitized anyway. So it was either 1 or 0 and summed up over the whole protein. So we could actually show that the best 6149 models, the top 10 can easily be detected. Then we did that for a few of these proteins and sent it for peer review. What do you think happens? Peer review feels like this. One reviewer says, okay, the second reviewer says, okay, but the third reviewer says, kill them. So that's what happened. So he's ready to gobble you up. So although we had seemingly good results and could screen high quality models from poor predictions, reviewers came back with a big list of complaints. And this was one of our bioinformatics journals. So they said, go back and make a machine learning method. This is not good enough. You need to analyze much more data because it's become data science now. It's no longer about understanding basic principles or anything like that. So we needed a protein quality assessment, which is not just binary, but a multi-class problem because we have models that span poor quality to high quality. It's like a continuum. Structure files are very large. So we need a fast machine learning method, a scalable and interpretable one. We'll have to be able to tell the biologist why our method is performing well, which part of it is doing well, etc. Avoid overfitting. So we thought maybe random forest is better in this other than SVM or NN. So we chose that, took two large data sets, CASP11 and CASP12. 70% was made as training and 30% stuffed as a test set. Now remember, for one protein, you can have many models that have been predicted by different teams all over the world and then submitted. So we had to choose the targets and their associated protein models. So we don't actually split the target. So we chose 38 targets and all their models and 15 targets here with all their models. That's roughly a 70-30 split. So this is what we do. We collect all the data, split it into two, and then try to throw it into a black box and then figure out what's happening and then try to figure out what it's predicting and why it's predicting that. So that's what is normally done. And so we wanted to do slightly better. So we'll show you how we did this. We also have blind test sets. We had two different sets that we used in this paper. And that's what we are looking at right now. The results will be shown to you because after testing, we applied that model to look at these test sets, which the method had not seen, and then we can evaluate how good our model is doing. Now, Einstein said, for feature selection, make everything as simple as possible, but no simpler. That means select the best features and add new features, but do not add too many. Keep it as minimal as possible. So that should be your aim. Choose the best features for training and then add only those that are essential. Try to minimize, because if you have too many features, you won't be able to explain your results. This is all supervised learning. We are not doing black box unsupervised learning. So of course, we started with the SAT score, so we have to use that. So that's one of our main features. But we also used protein secondary structure percentages, helix, sheet, and loop fractions. Loops are usually most flexible, so we thought maybe the percentage of loops, the loop content would be very important to understand whether a protein is disordered or it's an orderly protein, because that could skew some of our results. And then how much of the loops are contributing to our SAP score, because how much of the loops are hydrophobic, and that's what we wanted to see. And we checked for dependencies. These were all independent, so we are okay. The diagonal is really large, so we were confident that our features are fairly independent. That's very important, because if you have dependent features, it's going to skew your results. Right, so we used random forest. So that's finding the trees. So the data set is split binarily into a tree-like structure based on the values of the features. Then the two branches are analyzed for further subclassification. So it's two-way split. And we generated 700 trees. So that's what we have done. We calculated the PCC and we found the observed and predicted global distance template, which is a value that is used in quantifying two structures and how well they overlap with each other. We are taking a model and the true result, which is the experimental structure, to calculate GDT. We found that over 50 was a true positive and did three-way cross-validation and everything was consistent. So we were very happy. And then we thought, let's apply this to the blind data sets. We used the Cameo models. And then we got a reasonably good value, AUC of 0.82, which looks good. And this is like the blind test set. So as you can see, the blind man is trying to read. So that's what we were trying to do here, trying to understand what happened. Then we looked at domain-wise, how did we do? We seem to have done pretty well. We did really, and we compared to the original SAB CASP methods that were published, that are still online for CASP 12. You can see these results. And we found that we were doing quite well and even slightly better than some of these methods when we look at our very simple approach to life. This was published in Scientific Reports. After a couple of reviews, they made our life miserable, but then we are happy with the result. The moral of the story is never give up. Even a simple idea can actually do well. Avinash did the work. And he was in Australia at that time. He was in my lab for some time. Abdul is my colleague who was in Brisbane. And he is also from Rajasthan, by the way. Apparently he comes from a very prestigious noble family, maybe a royal family, I don't know. And Professor Jairam retired a couple of years ago from IIT Delhi. And he's like my batchmate. So we are about the same age. Australia, you don't have to retire yet. They are trying to make me retire, but I think I have to work hard. Now, in my own group, we looked at a totally different problem. Can machine learning be actually used on small molecules for drug design? Machine learning has been used for gene prediction, for microRNA prediction, for many of the other problems in genomics, transcriptomics, looking at gene expression arrays. We've also done a lot of work on proteins. So we wanted to see if it will actually work on the small molecule. For that, we are looking at the missing human proteome, which I worked with a colleague of mine who was president of HUPO here in Australia in my university. And this is of particular interest to him. Of all the human proteins, we know only this much. This much, we really have some inclination it might exist, but almost 574 proteins are possibly wrongly classified and they're probably non-coding. So our interest is in the missing proteins, which have some evidence at the transcriptome, homology, or prediction level. and are possibly proteins. And when we looked at the families of these, we found some of these names. This is just a word cloud. You can generate this yourself. And the size of the characters actually represents the number of members. And you can see what stands out are the olfactory receptors. It's a family of GPCRs, G protein coupled receptors, and it's the largest family of missing proteins. Now, what do these olfactory receptors do? They help you to smell, but many of them are also present in non-nasal or ectopic tissues. Ectopic expression means they are expressed in a place quite far from the nose, such as the brain, the heart, the kidneys, even prostate. The year Linda Buck and Richard Axel actually published the paper on olfactory receptors, they got the Nobel Prize in 2004. The same year, there was a paper that came out saying a similar receptor had been found in prostate cancer cells. So as you can see, they are involved in many things. They are involved in spermatozoa, kidneys, etc. And all the olfactory receptors seem to be doing totally different things. They are chemosensory receptors. They're actually looking for some molecule to bind. So that is the best suggestion that we could come up with. And it is well known that they are expressed in a huge array of tissues sorted by expression across all human tissues. 40 of the most highly expressed ones are shown here out of the 405. We have actually found a lot of evidence for many of these from wet lab, that means from mutational studies or binding studies, as well as from other associated studies. And we thought, let's take one and see if we can actually do some machine learning, because we have 143 compounds that are known to bind to this one particular olfactory receptor, OR1G1, which is expressed in enterochromaffin tumor cells in the gut. So our idea was collect all the little molecules, the compounds, the chemical molecules, do some descriptor, which is actually our fingerprint, which is like feature determination, and then process and clean the data, filter it, do some unsupervised and supervised learning, and then do some cross-validation, confusion matrix, estimate accuracy, sensitivity, do some prediction, and send for in vitro testing. In vitro testing is very difficult. There are only like 2 labs in the world which are working in olfactory receptors, maybe 3, 2 in the US and 1 in Germany. So we have to convince them that our values are very good. We managed to do that for another one because this one does not express according to our experimental collaborator in the US. So anyway, we are giving you the results. So we can show you that when you have so many molecules, we found then that there were other studies in the literature where they could find agonists using a similar method, but they used SVM, support vector machine, and predicted compounds were tested using a luciferase assay. Now this collaborator, the experimentalist in this group, is the one that we are working with, and we've submitted a paper which is under review. You know how paper reviews are, it's like a black hole, you have to wait forever. So we are waiting. So how did we do this? For small molecules, the story is very different. There is no amino acid here, there is no secondary structure. So we use what are called moderate descriptors for small molecules, small organic compounds. And these descriptors are generated using this GitHub freely available program. We have used all freely available programs in this study so other people can learn and use these different methods. And we have done the missing value filter, removed stuff that was missing. We have used the low variance filter, and then we have used the ranker, and we ended up with 13. There are 74 agonists and 74 non-agonists. So all the cleaning and pre-processing was done through KNIME, and the Orange software package. Orange is a machine learning package that is freely available, and it can be used by anybody. It has a graphical interface, so it's rather easy for students to learn and use. And these pictures were also generated with Orange. So we used three classical machine learning algorithms and evaluated each of them: SVM, RF, and naive Bayes, as well as a deep learning model, neural networks. So we used these four methods for the learning. Three are machine learning and one is neural network. So we made 125 trees in RF, and we use radius basis function for SVM. These are standard things. And then we use default parameters for naive Bayes. And we use 5-fold, 10-fold, 20-fold, and leave one out, cross validation, just to check how did our classifiers do. So after all this, we predicted a model, and then we used the predictions, and then we used we evaluated various measures to test how they did. So here are the classifications. We used area under the curve, the receiver operating curve, to estimate the overall performance. Then we looked at sensitivity, specificity, and F1. So when we did that, we found that these are requirements that we only did 10-fold cross-validation, but then the reviewers came back and said, that's not good enough. You have to do 5, 20, everything, and show us. So we have shown them everything. So the highlighted values are the best values. So as you can see, naive Bayes seems to be doing really well in many of these cases. So that's what we found. RF did well only in one case, where naive Bayes 20-fold cross-validation as well as leave one out looked really good. Naive Bayes in 20-fold did really the best. So we used that, and then we went for prediction. And for the prediction, we actually had to choose very large data sets, which we have done. So we, oh, sorry, I think we moved too fast. We used a human metabolite database from Canada, HMDB, which has about 114,000 compounds. We used CHEBI from EBI, European Bioinformatics Institute. This is the chemical database. And then we use the cancer odor database because we are dealing with odor receptors, olfactory receptors, and cancerous tissues, because tumor is what we were looking at. And there is another database called OdorDB. So we compiled and pooled all these small molecule data. And since the classifiers were trained on 13 selected features, the test data set was also described using the same features, so we can then mix and match. We used the Maxis fingerprints in KNIME to extract test compounds that were chemically similar. Since the data set is very large, we had to screen the number of positives, and then we selected those that matched our agonists and non-agonists very well, with a Tanimoto score of 0.85, which led us to only 1,500 compounds. And then we found that naive Bayes did the best in the earlier study, so we used that for the prediction. And naive Bayes actually predicted about 300 compounds that were really good as agonists. And since the predictivity, sensitivity of the predictive model was only this, we used a threshold to select the best predictions. Otherwise, we will end up with lots of false positives. So using that, we ended up with about 5 compounds. I'm not even adding well, I'm sorry. I've had a very long day today. So after these predicted compounds, all 5 have known, the 5 have known to have bioactivity are shown here. And we sent them for experimental testing, but our experimental collaborator could not express this receptor. He says it doesn't work for me, so he worked on something else. But we feel that we are actually got something, and if somebody can express this OR1G1, they can test out our predictions. So machine learning has lots of limitations, and especially for GPCRs, what we found was we need sufficient training data. Data science only works when you have large numbers of data. If you had less than the 100 compounds, things don't usually work very well. And you have to go for some other way for the drug design. So we use what is called pharmacophore modeling or virtual screening. And based on this, we have sent another lot of predictions which worked. And so now we have submitted the paper and we are waiting for the reviews. So additionally, machine learning approaches can only see in compounds that lie within the known chemical space. They have no way of actually predicting something that's totally novel. So these are limitations that we have to live with, unless we can come out with a new method that can break through this glass ceiling. So, but overall, we know that through machine learning approaches, we can distinguish between active and inactive compounds in a target selective manner and predict the bioactivities for drug discovery pipelines. These are very complicated systems, GPCRs, and only a few people seem to have the knack of expressing them. Saudamini has worked on olfactory receptors in insects, so she might be able to tell you a little more about those. This was published last year, and this year we are still waiting for lots of papers to come through. They are still stuck in reviewing. So I'm done. Thank you. I hope I'm on time, and I'm open for questions.